This video will be the first of 3 giving an overview of how to isolate and characterise commensal Staphylococci. Staphylococcus species are commonly opportunistic pathogens that cause sores, blisters, impetigo, boils, carbuncles, sepsis, and toxic shock syndrome. The problem pathogen MRSA is also a member of the Staphylococcus genus. Biomedical scientists commonly use flowcharts for specific identification of different bacterial species. A clinical specimen that is isolated as opaque cream or yellow colonies and that stains as a gram-positive cocci growing in clusters would be indicative of a Staphylococcus species. This could be confirmed with a catalase test, as almost all Staphylococci are catalase positive. Whilst a catalase negative result does not rule out a staphylococcal infection, other organisms should be investigated. A catalase positive result could be differentiated by a coagulase test, and common coagulase negative staphylococci include Staph epidermidis and Staph hominis, whilst the most common coagulase positive Staphylococcus species is Staph aureus. In fact, a suspected Staph aureus infection could be presumptively identified with a coagulase test. In this experiment, a primary swab will be taken from the skin and nasal passage, 2 common sites of staphylococcal carriage. These will be inoculated onto mannitol salt agar to obtain pure culture. A coagulase test will then be performed on the isolated species, and antibiotic sensitivity testing will be performed to identify the commensal staphylococci that are resistant or susceptible to certain antibiotics. To begin, take a sterile swab and roll around the inside of the nasal passage. Remember to make sure the environment is sterile using a Bunsen burner. Roll the swab across a small area of mannitol salt agar. The number of recovered organisms may be low, so remember to roll the entire swab over the agar. Take a sterile inoculating loop and streak out the plate from the inoculated area. Use the same technique you were shown in part 3.4 of the online lab book. Repeat this procedure using a fresh, sterile swab on the skin. These plates will be incubated overnight at 37 degrees Celsius. Mannitol salt agar contains a high salt concentration which selects for staphylococci. It also contains D-mannitol and phenol red, which allows mannitol fermenting and non-fermenting species to be differentiated. Fermentation produces an acid which turns the phenol red indicator yellow. After this incubation, we can see there has been non-mannitol fermenting growth from the skin swab. Choose one of the colonies to subculture onto fresh plates of mannitol salt agar to obtain a pure culture, and make a note of the colony morphology. Using a sterile inoculating loop, pick a single colony from the agar surface. Try to pick the whole colony without picking any neighbouring colonies. Use this to inoculate a small section of a fresh MSA plate and use the streak plating technique. We can see that there has been mannitol fermenting growth from the nasal swab. We will again make a note of the colony morphology and select a colony for subculture as before. If we take a look at these control plates, observe the lack of mannitol fermentation with Staph hominis and Staph epidermidis. Now observe the bright yellow colour of the mannitol fermenting Staph aureus.